Hello everyone, myself Dr. Parth Goswami, and today I'm going to teach you about intracellular accumulation, that is fatty liver. So first of all, let's understand the learning objective for the today's lecture. So it comes under the competency number PA 2.3, in which a student need to understand the intracellular accumulation of various substances like that of fat, protein, carbohydrates, and the pigment. So we will understand the definition of intracellular accumulation. Then I will teach you a mechanism of the intracellular accumulation. And finally, I will teach you intracellular lipid accumulation. We will discuss the accumulation of protein, carbohydrate, and pigment in another lecture. And obviously, at the end of lecture, I will discuss some of the MCQ. So first of all, what do you mean by intracellular accumulation? Definition. So the name itself suggests, it's a intracellular accumulation of either normal substance or abnormal substance. And it's due to the metabolic derangement. The cause is metabolic derangement. Because of abnormal metabolism, there will be intracellular accumulation. Now, this intracellular accumulation within the cell can lead to reversible type of cell injury. It can damage the cell, it can injure the cell. But suppose this accumulation is severe, then it can lead to irreversible cell injury. So both type of cell injury can occur because of intracellular accumulation. All right. Now let's see the mechanism of intracellular accumulation. So basically there are four mechanisms of intracellular accumulation of various substance. The first mechanism is abnormal metabolism of the substance. Second one is defect in the protein folding and transport. Third one is lack of certain key enzymes. And the fourth one is abnormal exogenous substance accumulation. Now let's discuss each mechanism in the detail one by one with an example. So the first mechanism of the intracellular accumulation is abnormal metabolism. All right. So the name itself suggests, here the metabolism is defective. The metabolism is not proper. There could be abnormal folding and transporting. Metabolism is defective. And that's why you can see that here there is a normal cell, right? But if there is an abnormal metabolism of lipid, then it can lead to fatty liver, in which there is a triglyceride type of fat accumulation within the liver. You can see a triglyceride fat accumulation within the liver, which is known by the name fatty liver. So the fatty liver is a most common example of abnormal metabolism mechanism. All right. The second mechanism is defect in the protein folding and transporting. Here there is a problem in the packaging, folding, and the transporting of certain proteins, and it can be seen because of mutation in the protein, right? Here there is a problem in the packaging, folding, and transporting of affected protein, and it's commonly due to abnormal mutated form of protein. The common example is alpha 1 antitrypsin accumulation within the cell. All right. The third mechanism is lack of key enzymes. Now see, this diagram is very interesting. See, this is the complex substrate, right? It will get degrade into soluble products, or we can say simplified products with the help of the enzyme. So enzyme will help in degradation of complex substance. So if you have if the patient lacking the key enzymes, then complex substrate will not get degraded and they will get accumulated within the cell. The most common example is lysosomal storage disease. Here there is a inborn defect in the enzyme. The enzyme is absent, you know so that can lead to lysosomal storage disease. We will discuss it in the detail in the genetics chapter, right? But just now, as of now, understand that if you lack the key enzyme, then complex substrate will not get degraded and they will get accumulated. The example is lysosomal storage disorder. The fourth mechanism is an abnormal exogenous substance accumulation. This is very interesting mechanism. See, suppose any patient exposed to carbon particles, if exposed to silica particles, if they have done tattooing over their body, then all these substances are exogenous for us. They are the exogenous substance. So if they get entry into the cell, then they are the indigestible material, because the body doesn't have any enzyme or any mechanism for the digestion of these exogenous substance. They cannot digest their substance, and they cannot even transport this exogenous substance. So there will be accumulation of this exogenous substance, which can lead to cell injury. The carbon accumulation is known by the name, coal worker pneumoconiosis. Silica exposure can lead to silicosis. Tattooing also an example of intracellular accumulation. All right. So today we are going to discuss about this. These all are the in general mechanism of intracellular accumulation. Now I will teach you intracellular lipid accumulation in the detail in this particular lecture. All right. So which lipid will accumulate inside the cell? Which lipid will get accumulate inside the cell, which is the example of intracellular lipid accumulation. So there could be accumulation of three type of lipid: cholesterol, phospholipid, and the triglyceride. These three form can get accumulated in case of an intracellular lipid accumulation. And the organs involved by fatty change are liver, heart, muscles, and the kidney. These four organs are involved commonly. So basically there are five main examples of fatty change, right? We are going to discuss about intracellular lipid accumulation, and this fatty change could be of the five variety. The first one is fatty liver, which is a very important topic. The second one is heart. Third one is atherosclerosis. The fourth one is xanthoma. Fifth one is cholesterolosis. And the final one is Niemann-Pick disease type C. All right. So first of all, let's discuss about the fatty chain, that is the steatosis. It is the most important short note. The fatty chain is a most important long answer as well as short answer question. So what is fatty chain? All right. So the fatty chain means there is an accumulation of the cholesterol, triglyceride, or phospholipid into the liver hepatocyte, right? That is the fatty chain. And here the most common form of accumulation of cholesterol is in the form of triglyceride. Triglyceride lipid accumulation in the hepatocyte is most commonly seen in the fatty chain, which is a most important MCQ. Which form of cholesterol get accumulated in the fatty liver? Then the answer would be triglyceride. All right. Sometimes lipid along with carbohydrate can get accumulated in the various cell in case of an lysosomal storage disorder. Lysosomal storage disease. Now let's see the etiology of fatty change. Why fatty liver occurs? See, the fatty liver is a very common problem nowadays in the community as well. Suppose if I am teaching 100 students, then if I do the ultrasonography of all the students, then 10 to 20% students might have fatty change. It's a very common problem. So you need to understand what is the etiology, why the fat accumulate in the liver. So obviously the causes could be of two types. One is if you have the increased lipid in your blood, which is known by the name hyperlipidemia, or you can have hepatocyte damage. If you have the hepatocyte damage, then lipid metabolism is affected, and so the fat is getting accumulated in the liver. So in which condition hyperlipidemia is occurring? So you can have high lipid level in your blood if the patient is obese. So the obesity is most important cause. The second one is diabetes mellitus. And third one is if you have the congenital defect. Congenitally also there can be hyperlipidemia. All right. Now, why hepatocyte damage? Which mechanism lead to fatty change? So, the hepatocyte can be damaged in case of an starvation, then alcohol, then protein energy malnutrition, then in case of an less oxygen supply, anoxia, then it can be seen in case of a certain toxin like that of chloroform, carbon tetrachloride. All these conditions can lead to hepatocyte damage, and so ultimately fatty liver. Another example is Reye syndrome, right? So all these are etiology for the development of fatty liver. All right, now let's see the pathogenesis of fatty liver, how it occurs. Pathogenesis means mechanism of the development of fatty liver. So before you understand the pathogenesis of fatty liver, you need to understand how the fat is metabolized in the liver. Why fatty change is common in liver? Because liver is the major organ for the lipid metabolism. So see how the lipid is metabolized in the liver. So you can get a free fatty acid from the diet, from the adipose tissue or from the diet, right? If you take it in the diet, then it will be absorbed from the intestine and will get converted into free fatty acid. So lipid getting converted into free fatty acid. And from the adipose tissue also you can get the free fatty acid. Now this free fatty acid, first of all, enter into the liver, right? After entry into the liver, they will be immediately esterified into triglyceride with the help of one substance. The name is alpha glycerophosphate. But a small portion of fatty acid can get converted into cholesterol ester, phospholipid, and ketone bodies. Understand? Now this triglyceride, with the help of apoprotein, lipid acceptor protein, they will convert it into lipoprotein, and this lipoprotein will get excreted in the plasma, will get released in the blood, right? So that is the metabolism of fat in the liver. Now, fatty liver can develop if you have the problem in any of these metabolic pathways, right? For example, if there is a hyperlipidemia in your blood, if you are taking excessive lipid in your diet, then there will be more free fatty acid and there will be more free fatty acid entry into the liver. Such free fatty acid entry into the liver can increase if patient is starvated, and if the patient is having diabetes, then from the peripheral tissue, the free fatty acid from the adipose tissue, free fatty acid will get entry into the liver and more fatty acid converted into triglyceride, and ultimately they will increases the chances of development of fatty liver. Second mechanism is that, if the conversion of fatty acid into these cholesterol, phospholipid, and ketone bodies decrease, then there will be increased esterification into triglycerides. So that is also one of the mechanism. Now, suppose if the patient is having protein energy malnutrition, then this apolipoprotein is defective. This lipid acceptor protein is not present. So there will be accumulation of triglyceride, because if this protein is not present, then triglyceride will not convert it into lipoprotein, and so the triglyceride will get accumulated in the liver. Understand? So any of this mechanism can involve in the development of fatty liver. Hope you have understand that pathogenesis, right? All right. Now morphology of fatty liver. How the fatty liver looks histopathologically. So this is very interesting. I will teach you in the image itself. I have taken this both image from the Hirschman pathology book, right? It's also a very good book. Ideal one is Robbins, but this book is also good. All right. So see, morphology means you have to write down the gross and microscopic appearance. So grossly, the liver is enlarged in size with yellow color, and it is soft, right? Enlarged in size, yellow color because of lipid accumulation, and soft. This is the microscopic appearance. So see, microscopically, initially, the fat will get accumulated around the nucleus, right? So it appear as a vacuoles, and it is known by the name microvesicle. See, this is the microvesicle. The fat is accumulated around the nucleus and it appears as a vacuoles. Now, why here the lipid is white color? You might have question. So the white color is because the fat will get removed during the automated tissue processing. That's why fat look white here, right? It will remove during tissue processing. All right. Now, suppose if the fat accumulation increases in the cell, then it will displace the nucleus to the periphery and more lipid accumulated in the cell, which is known by the name macrovesicle. See, this is the macrovesicle, right? The nucleus pushed to the periphery, you know, because the fat is increasing in the cell, so it will displace the nucleus to the periphery. Now, suppose still more the fat is getting accumulated, right? The fatty liver is not treated, and still the fat is getting accumulated in the macrovesicle, then it can lead to giant enlargement of the cell with only dot of nucleus at the periphery. And there will be lots of lipid in this cytoplasm, lots of large vacuole, which is known by the name fatty cyst, right? This is also the image showing fatty cyst and fatty change, right? These all are the fatty cyst. All right. So that's the morphology of fatty liver. All right. Now, fatty change in the heart. So, in the heart, usually fatty change occur in an altered pattern. Now, what is this altered pattern? Let me teach you in the detail. So, if there is a fatty change in the liver, fatty change in the heart in the altered pattern, then there will be two portion in the myocardium. One is involved portion by fatty change. Involved portion. An involved portion will look yellow color. Because of fatty change, it will look yellow color. While uninvolved portion of myocardium, will be red color. It's a red color because it is uninvolved. It contains the blood. So in the heart, if there is a fatty chain, then there will be alternate area of this involved portion and uninvolved portion, which will generate a yellow and red color bands, which is known by the name, it look like a tiger effect. It is given the name tiger effect, and the another name is tabby cat defect. This is the most important future MCQ. I am suspecting that this type of MCQ will be asked in your exam. Now, another example of intracellular lipid accumulation is a atherosclerosis, which is very common nowadays, right, in the USA countries and in the Indian country as well. Atherosclerosis means tunica intima, thickening by fat accumulation. Understand? Tunica intima is the first layer of blood vessel. Suppose this is your blood vessel, then this is the tunica intima. It is a endothelial line. Sorry, tunica intima. All right. Below which there is a presence of, smooth muscles. The smooth muscle layer is known by the name tunica media. Sorry, here I have written tunica intima, below which there is a presence of tunica media, and that is covered by adventitia. So the atherosclerosis means tunica intima thickening. The lipid is getting accumulated in this tunica intima, so there will be tunica intima thickening, and the accumulated lipid is cholesterol type or cholesterol ester. And here the lipid is getting accumulated in a two cell, macrophage and smooth muscle cell. These two cells will contain the lipid within it. Yes, don't get surprised. The smooth muscle will also imbibe the lipid within it. The macrophage is having the scavenger receptor by which they can imbibe the lipid within it. So macrophage and smooth muscle cells will take the lipid within it, and there will be intimal thickening, that is atherosclerosis. And ultimately, the atherosclerosis commonly involve the aorta and its branch carotid, sorry, coronary artery. They will involve the aorta and its branch coronary artery. So finally, if coronary artery is involved by atherosclerosis to a severe degree, then it can lead to ischemic heart disease, that is the heart attack. All right. Now xanthoma, another example of intracellular lipid accumulation. So what do we mean by xanthoma? Oma means benign tumor, right? The suffix oma is given for benign tumor. So it's a benign tumor of macrophage that is filled with lipid. Remember this word. The macrophage filled with lipid will form a mass, will form a benign tumor mass, which is known by the name xanthoma. This macrophage in the xanthoma is known by the name foamy cell, and it commonly get accumulated in the skin and tendons, right? It involves skin and tendons commonly. All right. Now cholesterolosis. See, these are the cholesterol-laden macrophages. See, this is the macrophage. Peripherally pushed nucleus and a clear vacuole within the cell. These all are the foamy cells. Macrophage. Foamy macrophage. This cholesterolosis is seen in the gallbladder. It's seen in gallbladder. We know very well that the gallbladder histologically having four layer: mucosa, submucosa, muscularis propria, and sedosa. Now, in between these mucosa and submucosa, there is a presence of lamina propria, right? So, if in the lamina propria there is an accumulation of this foamy macrophage, then this condition is known by the name cholesterolosis, right? So, this is this condition. There is a presence of foamy macrophage in the lamina propria. All right. The last example of intracellular lipid accumulation is Niemann-Pick disease type C. It's a lysosomal storage disorder. Lysosomal storage disease. Here there is a defect in the enzyme. There is a lack of enzyme which involved in cholesterol metabolism or cholesterol trafficking, right? So there is a defect in the enzyme that involved in cholesterol trafficking, and so that's why there will be lipid accumulation in many organs, right? In many organs. So that's the type C Niemann pipe disease. All right. Now let's see some of the MCQs. Okay. So clear, these all are the example of intracellular accumulation, right? Fatty liver, fatty change in the heart, atherosclerosis, xanthoma, cholesterolosis, and Niemann pipe disease type C. First MCQ, 42-year male with a chronic alcohol having the drinking habit has elevated SGPT level in the blood. The liver biopsy histopathology image attached herewith. Which of the following change best reflect this condition? So first of all, you need to diagnose this condition. This is the fatty change. Can you able to see a fatty cyst and microvesicle? So, this is the fatty change. This is the fatty liver condition. So which change best reflect this condition? Fat necrosis, apoptosis, triglyceride accumulation, or protein misfolding? So commonly fatty change is because of triglyceride type of fat accumulation. So the answer is triglyceride accumulation. This is a most important MCQ. Such type of MCQ can be asked in your exam. I have prepared this MCQ especially for you. 44-year male developed grade 3 fatty liver due to prolonged starvation. Which mechanism could be responsible for the fatty liver? Increase free fatty acid entry into the liver, increase acetate conversion to phospholipid, increase protein degradation, or apoptosis. So here the answer is C. In case of an starvation, there will be increased free fatty acid from the periphery, and if there is an increased free fatty acid into the liver, then it will esterified into triglyceride, which will get accumulated in the liver. So the right answer would be the increased free fatty acid entry into the liver, right? So this is all about the intracellular accumulation. Hope you have enjoyed this simple video. I will be right back with a new video. Till then, take care and bye bye. Thank you.